Good morning or afternoon, class. I wanted to finish out the notes on DNA RNA unit. Hopefully you've already watched the video and learned about chromosomes, about DNA replication, and transcription translation. Basically how you go from DNA to mRNA in transcription to then translation, which takes the mRNA and makes a protein. If I could get you to pause the video after you open a new window and go to the slideshow for the notes. I am on slide number 39. So on slide 39, it has a picture of protein folding. That's what happens after we make our protein from transcription translation. All we did is we created a long thread of amino acids, and we need to fold it and weave it to create it into an actual 3D shape, which our proteins normally do have a 3D shape. They could look like a pump or a channel, and some of them are very long, like fibers. The spindle fibers used to do cell cycle, breaking the chromosomes apart in anaphase is a protein. It's made of fibers, a bunch of amino acids all chained together like a long rope. On slide number 40 is actually where I wanted to start. So mutations. Mutations can happen in the DNA sequence. We can have some changes that can happen by accident. It could happen because the cell made a miscopy in copying the DNA in DNA replication, or it could be from some sort of outside factor, like for example, a chemical that is absorbed by the cell that mutates the DNA, or it could be energy, could be like radiation. If you're laying out in the sun, getting sunburnt, all that radiant energy coming from the sun, UV light, can damage our DNA, can cause mutations. So on the slide, there is a sentence in bold that's not too hard to read. If every word is 3 letters, it reads, "The sun was hot, but the old man did not get his hat." Easy enough. Until, if you go on the next slide, number 41, you see that all we have to do is insert or delete 1 or 2 letters, then the whole sentence no longer makes sense. And this happens with our DNA occasionally. But we have enzymes that go back and proofread and make changes and fixes based off of what's needed. The younger you are, the faster they are at doing this. But once you get older and your immune system wears out and your cells are not as active as they used to be, then mutations can start to build up. And those mutations can wreak havoc on our cells. It can lead to something like cancer. That is unfortunate, but we do have fail-safes. We have our cells trying to fight against that. And if there is a large mutation, our cells are very good at usually spotting and defeating it. On the next slide, 42, some consequences of mutations. So mutations, if it happens in a multicellular organism, it will stay in that organism unless it is a gamete cell. If it's a gamete cell, then it could get passed on to the offspring. A gamete is a sex cell. For humans, that's our sperm and our egg. But if it's in a multicellular organism that's not going to be our sperm and egg, like you and I, then it's just a mutant cell. And that mutant cell can go through cell cycle and make 2 mutant cells. And those 2 mutant cells can make 4, and then 4 to 8, and it keeps dividing and dividing and dividing. If they are harmful, then we consider them cancer. Now, the next slide, which is 43, there's the note about gamete cells, sex cells, that can be passed down to the next generation. Some mutations that we looked at in the human genome, our genetics, are related to some diseases, such as sickle cell anemia is 1 base pair. I believe it is an A and a T. They're in the wrong place. Or it should be an A and T, and it's something else that causes a misfold of hemoglobin, the protein that's in red blood cells that make the red blood cells nice and round. Instead, they end up in a crescent shape or a bent, kind of like the moon on the South Carolina flag. Taysher's disease, such as fibrosis, hemophilia, Huntington's disease, these are all related to a miscopy or mutation in the DNA. Next slide, which is 44. Not all mutations are bad. Some mutations can be beneficial. They don't happen as often, but we do have documentation of them existing. Like, for example, my favorite example of a beneficial mutation was discovered, I believe, in southern India. and it is a mutation of a protein that's found on our cell membranes. That protein has a name, but its name is very long, so we shorten it with the abbreviation CCR5. The CCR5 protein is a channel that allows things to come in and out of the cell. And with a mutation, it doesn't allow everything to come through like it normally does, and that sounds like it's a bad thing. But with the mutation, it blocks the HIV virus from coming in the cell. So if you have that mutation, then you are less susceptible to being infected by HIV. This is a beneficial mutation that some people inherit. It gets passed down from father or mother to child. Now, of course, you get 2 copies of these genes. You get one from your mom and one from your dad. So you could inherit 1 copy from one parent and the normal copy from the other parent. So you're only partly immune to HIV. Or you could inherit a mutated form from both parents, and then it appears that you're completely immune from HIV/AIDS. It just depends on what you inherit. Now, beneficial mutations are out there. They're just not as common as harmful mutations. If you go to the next slide, it is slide number 45. 45. I go into viruses a little bit. But if you wanted to read about mutations, they're in the textbook page 238 through 241. If you want to read about viruses, it's in the textbook page 513 to 517. So viruses can wreak havoc on our cells and manipulate our DNA RNA. So I have my whiteboard. A virus is made up only of a few things. This is a weird drawing one. They come in different sizes, different shapes. I just made one up. Inside a virus is either DNA or RNA. Viruses don't have both. It's either a DNA version or an RNA version. HIV/AIDS is a DNA virus. So is influenza. The current coronavirus is actually an RNA virus, and so is rabies that causes fear for dogs. They start foaming in the mouth, they're rabid. So that's an RNA virus. Inside the virus, there is one of those 2 nucleic acids and a bunch of enzymes. What's holding it together is some proteins. We call it a protein coat because it coats over the nucleic acids. And sometimes it uses a phospholipid bilayer. Now, viruses need to be able to get inside the cell. Some of them are very, very small and might be able to slip through a channel, but most of the time they're bigger than a channel will allow to get into the cell. So the cell actually has to pull it in through a process called endocytosis. But if they're going to get pulled in, they have to be welcomed. On the outside of our phospholipid bilayer, our cell membrane, are a bunch of proteins. Some of them serve the job as pumps and channels, but some of them are also signal listeners. They wait for signals from the outside for something to be sent from another cell with a label that says, bring this in. So here is a protein on the outside of the phospholipid bilayer, and it's got a unique shape created by the process of transcription, translation, and folding. And once the virus binds to it, if it binds to it correctly, then the protein will be pulled into the cell membrane. Along with it will be the virus. The virus has the key to get inside the cell. It used the right key. Now, if it is the wrong key, if it doesn't fit, then it can't get in the cell. There are a lot of different viruses, thousands and thousands. Some infect plants, some infect bacteria, some are specialized for attacking bacteria. We call those bacteriophages. So currently right now on your skin and your hair, there's probably dozens and dozens of viruses, but don't panic because most of them can't get in your cells. They attack other things like plants and fungi and bacteria. We have to have one that has the right key to attach the protein to get into the cell. Once it gets in the cell, then it can hijack the process of transcription translation. If it is a DNA variety of virus, it'll insert its DNA into your nucleus, and then it'll get that DNA transcribed, and then translated into new viruses. If it is an RNA virus, then it can skip that whole getting in the nucleus, and it can go ahead and start inserting its RNA into your ribosomes, and the ribosomes will start building new viruses. I'm going to move forward onto slide 46. Viruses cannot copy themselves on their own. They have to have a cell. This is why we don't consider them living, along with other things. They need help. They need the machinery of the cell to do this. So they get inside of a cell, and then they start using your enzymes and your ribosomes to make copies of itself. I'll also post a couple of videos so you can see how viruses do this, because it's a little bit easier to see this in animation version. But once the virus is inside, it can copy itself over and over again, filling up the inside of the cell with a lot of different viruses, a lot of copies of itself. And then once it gets too full, it'll burst the cell open and release those viruses to go infect new cells. Those viruses can also travel through the lymphatic system. It can travel through our veins and arteries to find new cells to attack. Viruses can be very contagious, but your body is trying to battle against them. Eventually, your body should learn that the virus is something harmful, will recognize those proteins, those sequences of DNA or RNA as harmful, and will start attacking them. This is not instant, it takes some time, but the good news is your cells have memory. You may remember at the beginning of the class when we were covering a little bit about smallpox. There's a vaccine for smallpox that helped eradicate it. And when there was an outbreak of smallpox, they would vaccinate everyone in the surrounding towns around the outbreak. So then the outbreak couldn't spread any further, kind of like surrounding a fire with a fire break, with tilling the soil or saturating the land around it, so the fire will just burn out and can't get outwards. But vaccines are slow to make usually, but it's a copy of the virus scrambled. So it's not a strong version, it's a very weak version that we've scrambled, we've mutated, so it's less harmful. And what we do is we basically infect you with a vaccine. We put a version of the virus in you that's very, very weak, and that gives your body a chance to go through a checklist. It goes through a checklist to see, all right, will this destroy it? Nope, that didn't work. Go to number 2. Will this destroy it? Nope, go to number 3. And we'll start going down through a checklist to figure out how to defeat and break down that virus. Once it finds something that works, then it's going to make a note. It's going to note that in the nucleus what was successful for that virus, and then your body should be fairly immune from it in the future. Once that virus is exposed back to you, your body is going to be like, oh, I remember this, let me check the database. And those things didn't defeat it, but that did. So that's what we're going to use this time from the start. That's how your body fights viruses that you're exposed to again and again. There are some exceptions to this, like for example, you may have heard of chickenpox. If you have a person that suffered from chickenpox as a kid, they can have that virus mutate again in their adult years and start popping up again in their cells as a form of called shingles. And then your body has to deal with that mutated version. You may have also realized that the influenza or the flu virus comes in many different forms. There's a whole family of them. And just because you are vaccinated against type A doesn't mean that you're vaccinated against type D or E or F. So there's so many different types of the flu virus that we can't vaccinate everyone from every single strand, and the flu mutates pretty fast. So we just fight it one strain at a time each year. On the next slide, which is number 47, there's some questions. I'll answer them. Describe a double helix. That's referring to the DNA. Double means it's got 2 sides, and a helix is a spin or coil. That's the DNA molecule. What is a nucleotide? A nucleotide is the simplest, smallest unit of our DNA or RNA molecule, and it's made of 3 components. It's made up of a nitrogenous base like adenine, thymine, guanine, cytosine, or uracil. It's made of a sugar, And if it's DNA, it's deoxyribose sugar. If it's RNA, it's ribose sugar. And then it's also got a phosphate group. Those three components, and you can see that earlier in the slideshow at the top, is what our nucleotide is. Where are hydrogen bonds found in DNA? Well, that's what attaches the A to the T, so they hold together, or the G to the C to hold those together. Those are the hydrogen bonds holding the two nitrogenous bases. How do uracil and thymine relate? Well, both of them want to bond to adenine. Uracil is used in RNA to substitute for thymine, which is found in DNA, and both of them want to stick to adenine. Number 5, what are two differences between DNA and RNA? Well, there's more than two. For example, different sugars. We use thymine in DNA instead of uracil, which is in RNA. DNA is a double helix. It's got two sides. RNA is a single strand. DNA is stored in the nucleus of our eukaryotes. RNA is created in the nucleus, but is then sent out of the nucleus to be read by ribosomes. Our DNA is for long-term storage of information. Our RNA is for short-term storage of information before it gets broken down. All right, I hope this video helps, and I'll post a couple other videos along with it. Use your textbook, and I'll hope to hear from you soon.